We're going to go ahead and get started. Welcome to Medicine Grand Rounds. Just a few announcements before we start. We had a wonderful Shark Tank morning presentations, and I want to invite everybody to come to the poster sessions at 2 o'clock, and also the award ceremony at 4 o'clock in the afternoon. So everybody's welcome to attend to this. And before, oh yeah, we'll also, Dr. Weiss, remind me about the important part of it. So we'll have refreshment, including wine and beer. So everybody's invited. Before I let our chief resident introduce the speaker, I want to thank Dr. Snyder for accepting the invitation. You're going to see his work, and if I can define his research, I can say that this is transformative. You will see that his lab and his research group works on integration of omics, genomics, metabolomics, epigenetic changes combined with wearable information, and this will help to not only to predict disease but also to design strategies for personalized medicine. When you're going to see his presentation, you can see that he breathe and live data. You can see only that when you see his hands. And I want to thank for his, making an effort, especially this time, to come to Miami. And I'm just going to let our chief resident, Dr. Noy, to introduce him. Thank you.
Good afternoon, everyone. So today we're introducing Dr. Michael Snyder, who's a Stanford Ascherman professor and the director of the Center for Genomics and Personalized Medicine. Dr. Snyder received his PhD training at the California Institute of Technology and carried out postdoctoral training at Stanford University. He's a pioneer of and has invented many technologies that enable 21st century healthcare, including systems biology, RNA sequencing, protein chip technologies. He has led groundbreaking research in genomics, biotechnology, and personalized health, with a focus on how genetic variation and lifestyle affects individual health and disease, heralding a new and more effective approach to healthcare. He also studies how people age, demonstrating that we all age differently and that in general there are bursts of changes at age 44 and 60. He is known for his pioneering study, where he was both a subject and researcher, which opened new pathways in the understanding of personalized medicine and laid the groundwork for precision health. He is one of the first researchers to gather petabyte of data on individuals, which is 1 million to 1 trillion times more data than the average clinician collects. He has published over 1,000 papers and is one of the most cited scientists. Please welcome Dr. Snyder.
Okay, it is a real pleasure to be here. I'm enjoying the visit so far, and the Shark Tank was amazing, hearing all these great ideas. I'm going to talk about deep data for trying to transform healthcare, and I'll start with what motivates us, which is we think the healthcare system's broken. It's broken in that we tend to practice sick care rather than healthcare, and the consequence of this is that we tend to keep people alive versus healthy. And so it depends how you want to quantify this, but health span, let's see if we can get this working, here we go, is generally shorter than one's lifespan. And as I say, it depends how you quantify this, but that's about 11 to 15 years, meaning people are living their last decade of life with chronic disease or unhealthy. And obviously, I think most of us would like to think we should keep people, healthy all the way to the end, make this a rectangle so that people will, live healthy and then pass away. And probably at the same time, you'll extend lifespan as well. So also, if you think about it, even when we practice healthcare, it's pretty antiquated. You typically get in a car, always at an inconvenient time, to travel to a physician's office, which hasn't really changed that much in the last 40 years. There are some new technologies there, but compare that to the dentist's office, where you can get same-day crowns, the things that move very, very fast in dentistry, whereas in medicine, although they move fast maybe downstream on the treatment side, not as fast actually on the actual health care side, I would argue. And then while you're there, they'll obviously take lots of blood. And from all that blood in the time you're there, they don't really make all that many measurements when you think about it. And then from those measurements, they'll treat you based on population averages. And so we're trying to change all of these steps. And just with regards to this last one, from a layperson standpoint, you're told since you're little that your temperature is 98.6. But if you actually look at the data that's out there, it's not 98.6, first of all. Here's a study of about 3,000 people. It's more like 97.5. I myself am 97.3, and I've been dropping over the last 10 years. And we can argue what that means, but the point is that it's not 98.6, but the more important point up here on the slide is that it's a spread. Whereas the bottom of that box is a 25th quartile, 94.6, and the top is 75th quartile, 99.1. So what that means in today's world, if your normal healthy baseline is 94.6, you're measured in a physician's office at 98.6, they'll tell you you're normal, you're healthy, everything's fine, but I guarantee if you're up 4 degrees over your baseline, you're not healthy. And that's the big theme of our work. Know what your healthy baseline is so you can detect shifts. Now, your health, I think it's all influenced by many things. Your DNA is important. Anyone know how much your DNA contributes to your lifespan? How many say it's 50%? How many say it's 80%? Couple. How many say it's 20%? Yeah, it's actually 16%. There's a big error bar on that, though. Meaning all these other things like exercise, food, mental health, environmental exposure have a bigger impact. Now, it's true if you subtract out violence and other things, then genetics becomes 50%, although some of that may be due to other factors as well. Anyway, the point out of all this is that there's still a lot of room for lifestyle and for environmental exposures. And we're in a world where we can now quantify all these things. Some well, like activity, you can measure quite well. Food is clunky. Mental health, you can actually measure indirectly. And environmental exposures, we actually have a number of projects measuring that as well. But even if you don't measure these directly, you can measure their effects by doing deep profiling. This is something we started now about 16 years ago. Actually, on me, I'm one of the subjects. And then for the bulk of people, it's started with small group people, 109. We've been running this now for a little bit over 13 years. So the slide's a little bit dated. And what we do is we collect blood, urine, stool, and other actually samples from microbiome, and we'll do very, very deep measurements on those. We'll sequence their genome once, but then we'll measure the epigenome. So that's DNA methylation on a subset of people. It's pretty expensive. We'll measure transcriptome and proteome out of peripheral blood monocyte cells, so they're immune cells. And out of plasma, we'll also measure proteins, cytokines, metabolites, lipids. And we measure the microbiome out of stool, nasal, tongue, and skin. And that's quite interesting, actually. But for the purpose of this talk, I'll just talk a little bit about gut microbiome. That's on top of deep clinical tests, questionnaires we take, as well as advanced tests like stress echocardiograms, variety of glucose control measurements, because we're very interested in type 2 diabetes, which I know a lot of people here are interested in. And then about 11 years ago, we brought on the wearables when they came out as fitness trackers. We thought they'd be powerful health monitors. So we've integrated that. You'll see that's going to be a big theme in today's talk. And so one aspect of all this is to collect very, very deep data on people. And the other aspect is to do it longitudinally. So we'll sample people every 3 months while they're healthy, and then take 5 to 7 more samplings when they get ill. So you might say, why are we doing all this? Well, the answer is we're trying to understand what a healthy profile looks like, not a sick profile. How does it change over time? What happens when people first get ill? What's the difference between you and the person sitting next to you? And then one big theme relevant to, I guess, this group is that can we use advanced technologies for being able to better manage people's health? And I can tell you when we started sequencing people's healthy genomes back 13 years ago or something, most physicians were saying, that's a terrible idea. You're going to turn everybody into hypochondriacs. It's going to cost millions of dollars. This is not a good thing to do. And some were quite angry. But of course, we did it anyway. And I think most people have warmed up to this now, that you can actually learn things about disease risk. You'll see that in a minute. Most still don't like whole body MRIs or wearables. We'll get into that a little bit here as we go along. But anyway, so we do collect deep data on people. And then, as I say, from what we've learned just from the first 3.5 years, was that we had a number of major health discoveries, all pre-symptomatically. And they spanned all these different areas like hem-oncology, cardiovascular disease, metabolic disease, and some were a pretty big deal, meaning we caught someone with early lymphoma, two people with pre-cancers, one of which has now turned into aggressive cancer, this MGUS. Two people had serious heart issues. One was picked up by genome sequencing, another through wearables. And that was another lesson. No one technology found these. Sometimes it was the imaging that we had done. We did a limited amount of that. Sometimes the biochemistry from the omics. Sometimes it was the genome sequencing, actually quite frequently. And the idea is that what we're doing is we're getting a much more complete picture of people's health relative to what we do today. So if your health is a 1,000-piece jigsaw puzzle, we like to say we're trying to get as many as possible, say 700 or so, versus we would argue get more like 10 today. We think you can get a much, much deeper picture, and I'll come back to this theme a little bit later. Now, genome sequencing was quite powerful, and these days you can interpret genomes reasonably well by these single gene mutations. Many of you may know this. So these are called Mendelian mutations. And a prototype of this is, a classic is BRCA mutations. And one of our individuals, so from the first 70 people we sequenced, we have more now, one individual did have a BRCA mutation, so they're getting screened more. So 12 had very actionable information, and 3 of them, it turned out, had phenotypes associated with this, meaning this individual was a young fellow, had a mutation in RBM20 there, which is involved in cardio, if you will, function. And he actually did a follow-up, and sure enough, he actually has a heart defect. And that was picked up through this, and he's on medication now. His father actually died of a heart attack, and this is likely the culprit. Another individual, the top one up there, SDHB, there were 2 individuals who had a mutation there. They're at high risk for certain kinds of cancer. One of them did a whole body MRI follow-up and had early thyroid cancer that was removed, and they kept most of the thyroid, so they don't need hormone replacement. And 9 individuals coming in the study were thought to be type 2 diabetic. One of them turned out to have a MODY mutation. It's a different form of diabetes, many of you may know. And they actually switched their medication to something more effective as a consequence of this. So the point out of all this is that even though it's not the majority of people, it's a decent number of people had mutations here that was quite actionable. In some cases, we might argue potentially life-saving. Now, this is the Mendelian cases. I'm going to dive a little deeper into genetics. So if you don't like genetics, you can fall asleep for a little bit, but try and get the gist of this anyway. So single gene mutations, they're the easiest to interpret, and that's what people usually do from a genome sequence these days. But most diseases are complex disease, and these are thought to be due to common variants, common differences we all have in our genome. We have about 4 million of these, that if you add them up, if you have the bad variants, if you will, you add them up and you got a lot of bad variants, you're at higher risk. If you have the good variants, you're at lower risk for disease. And so the way people, so it's kind of thought like that would be killing you by death by 1,000 cuts versus single gene mutations maybe getting clobbered by a sledgehammer. And so the way people find these common variants associated with disease, you may know this already, you do these GWAS studies, genome-wide association studies, where you map cases and controls and look for signals that can find loci, genetic loci associated with the disease. And that's how you find these variants, and then you can build polygenic risk scores. So you can sum over literally thousands or millions of these variants and make predictive models about someone's genetic risk. The problem with this is it only works for about the top 2 to 4% of people. People are at the extreme end of the polygenic risk score. If you look at something called odds ratios, it really only works for that top group. For most people, it doesn't work. This is a very controversial area, by the way. So we think there are 2 flaws with this. One is it ignores all the rare variants that might contribute to disease. And the other is it's a linear addition. And anyone who does genetics knows genetics isn't linear, meaning if you have 2 mutations in the same biochemical pathway, 1 + 1 is 1, right? They don't add up because you're just killing the same pathway twice. If you had two mutations in different pathways, 1 + 1 can be 15. It can be very synergistic. So that's the problem with these. And so we've come up with new ways using, of course, AI to better get at the genetic basis of complex disease. We started this before it became quite so popular. It doesn't use LLM models at all. And I'm going to walk you through some examples of this. We have somewhat more sophisticated ones. I'll sneak that in at the end. But basically, we've taken on some of these complex diseases that are known to be quite heritable in the case of the top two. And these AI approaches, if you will, they find 10 to 100 times more genes typically than is normally found by traditional methods. So I'll walk you through the first way. So this is abdominal aortic aneurysm. Whoops. Sorry. I've got it. Yeah. Which is known to be highly heritable, and it kills about 10% of people over 65. So it's fairly common. Yeah, basically the way you typically find you're at risk for it is your aorta bursts and you die. So obviously we need a better system for being able to screen and track this, and we thought maybe the genetics would help get at this. So what we did was we sequenced just 268 cases, 133 controls. We did whole genome sequencing, but we zoomed in on the genes. So this is all about rare variants. And what we did was we could take the essentially deleterious pathogenic mutations in the genes that are either known or predicted. And then we also could add in some EHR data. And then you can do machine learning. Now, the power of machine learning is it's not linear. It can grab groups of genes that are enriched in cases versus control, and it's done in an agnostic fashion. You can do what's called tenfold cross-validation. And the bottom line is at the time we started, there were 3 genes that were kind of questionable. We found 60 genes involved in AA, and they wind up sitting in networks where you would expect AA to be operating, meaning they wind up in genes involved in blood circulation, blood pressure, cardiomyopathy. And I won't go through all the data, but mouse models of these genes give circulation or A kinds of defects. And the genes tend to be lower expressed in both patients and in mouse models. And then lastly, we could go on to build a polygenic risk score that's fairly decent. It is the case that, especially later in life, the EHR data does have some reasonably predictive value, but with the genome, you can do better and get rid of a lot of false positives, actually. So we think that we can get to at least some level of prediction on a complex disease with this kind of model, and I think we know how to improve this further. So that's the rare variant case. What about common variants? So here we're going to dig in a little deeper. I'm going to walk you through this with the case of ALS. Now, ALS, you may or may not know, it's highly heritable, actually, about 61% heritability. Even the spontaneous forms have a heritable basis. And here we're going to use GWAS data. Now, GWAS signals usually sit in regulatory regions. So we did 2 things here. One is we mapped out the regulatory regions of motor neurons, which is where ALS is thought to primarily hit, although I think it goes beyond motor neurons. So you can map out the regulatory regions with methods called ATAC-seq and ChIP-seq and such. And there's 100,000 of them in motor neurons. And then you could do the exact same principle, which is use machine learning, where you take GWAS signals, in this case, from about 12,000 cases and 23,000 controls. So that's a very large GWAS study for ALS, biggest at the time. And you do the same thing for each region. You say, is it enriched in cases versus controls? It's computationally very, very intensive, but it works. This GWAS study had found 7 genes, later 15, when they boosted it. We found 690 genes from the same study, okay? And in fact, we went from then 6% of the heritability, for those who follow this sort of thing, complex disease, you often don't know much of the heritability. We boosted it to 36%. And we did find a lot of the well-known ALS genes. A few others fell just below the threshold. And many of the other genes were enriched in things known to be involved in ALS, meaning by gene expression or other studies. And the genes are downregulated in the brains of ALS patients. as well as in iPS-derived motor neurons, iPSC-derived motor neurons. And in fact, the genes tend to operate in the distal regions of those motor neurons. And then we even took this one step further, where we could actually take iPS cells, and we thought one of the top genes might have a Mendelian form. It's called KANK1. So we went in and we actually regenerated that mutation in iPSC motor neurons and then differentiated them into we generated iPSCs, then generated motor neurons, and sure enough, they have ALS kinds of defects, including mislocalization of TDP43. For those of you who follow this, I know this is getting into the weeds probably a little bit, but this is thought to be a hallmark of ALS. And in fact, we could show that when we knocked this out, TDP43 mislocalization occurred, which says that there are things upstream of TDP43 that are involved. That turns out to be a bit of a big deal because everyone thinks that TDP43 is causative, which it may be in part. Anyway, the point out of all this is that we can find new genes and better explain the genetic heritability of ALS. And these days, we've even used this gene list to look at patients, and we can actually see likely causative mutations now from this gene list, which was not possible before. So we think this information will become very, very valuable. So that was nice. We can find the genetic basis, something where we know what the cell type is. What if you don't know the cell type? Okay. So, and the example I'm going to show you here is COVID severity. So during the, well, I guess we're not over the pandemic, we're the acute phase of the pandemic, I'll put it that way. There was a lot of interest in trying to get the genetic basis of COVID. So people over 65, can get severe COVID and die or wind up in a hospital. And that's because you tend to hit immune decline as you hit your 60s. But young people can get severe COVID and wind up in the hospital and die. And that's thought to have a genetic component, right? Anytime young folks get something, you think there might be a genetic basis for it. So there had been a really large GWAS study done of 5,000 cases, 1.4 million controls, to map out the loci responsible. And they'd found about 40 loci involved basically in severe COVID. And so we weren't sure what cell types were involved in severe COVID. So we took lung tissue where there's 19 cell types, and someone had done something called multiome, which is single cell RNA and attack-seq. It's a way of finding the regulatory regions. And sorry if there's too much in the weeds. I'll bring you back, though, on board later. But the point out of all this is for each cell type, we know the regulatory regions. There's 100,000. And once again, you can map the GWAS signal from that large study onto those cells. And you do machine learning and say, give us all the regions that are rich in case versus control. Once again, very computationally intensive. But it does work. We found over 6,000 regions mapping to over 1,000 genes across the various cell types. And in the end, what we could say is most of the signal, the heritability, sits in natural killer and T cells. We could even do further analysis and show it's a specific kind of natural killer cells. This is quite nice because at the beginning, some people were saying, well, most of genetics is in epithelial cells. It wasn't really clear where the genetic basis of severe COVID was. And we would say it lies primarily in these two cell types. And one of the things that was pretty spectacular, which was that because it was a very large GWAS study, for the most severe forms of severe COVID, we could explain 77% of the heritability, which is like unheard of for complex disease. So bottom line is we think with these new approaches, we can get much, much deeper at the genetic basis of complex disease. And there are ways to improve this further. We had a paper come out recently. Won't spend too much time on this, but you can take this one step further where you can go into these individual cell types, develop, single cell polygenic risk scores, and then you do something called graph networks that gives you boost and signal, if you will, where you boost across networks rather than individual genes, where you try and look for signals that way. And the net result is with this boosted extra signal, you can start, again, getting not only at the complex, the genetic basis, but also the cell types involved. And I know there's people interested in diabetes here, so I put the slide in. And the bottom line is by doing this new AI method for type 2 diabetes, which you may know there's been very large GWAS studies run, we can actually drill in, and these are the cells of the pancreas. There are many cell types here that have been identified by single cell. And we can map the genetic signal onto these, and most of the genetic signal sits kind of evenly between alpha and beta cells, the producing cells, meaning that alpha cells have a genetic basis for type 2 diabetes that hadn't really been appreciated before. I think people have certainly implicated alpha cells in type 2 diabetes, but not as a genetic basis. And I think our data says they probably do have a strong genetic component. And so we can even take this one step further by looking through the genes that we identified. We do find, again, genes involved in alpha cell function, those of beta cell. And even though this has been really well trodden by GWAS studies, half the genes we find in each of these cell types are new. They hadn't been identified. So the point out of all this is that we think these AI approaches are very, very powerful for getting a genetic base of complex disease. And I know there's a lot more room for improvement here. Now, I'll point out we're not yet strongly predictive for ALS or severe COVID just from the genome sequence. We can predict prognosis for ALS, how well people do, meaning is it severe or is it more mild form, but we haven't yet predicted well who's going to get it or not. We're still working on that. We may have to add other kinds of information. So anyway, that's the genetics. So if I lost all of you, don't worry, we're away from the genetics, but you get the idea. I think we can now get at genetic basis disease just much, much better with these new tools. Now, the other thing, because we're tracking people over time, we can see how they're changing. And if we just look at the healthy time points, we can see what pathways are changing, which we interpret as aging. And what we discover is that everybody ages differently, meaning this person here, their top biochemical pathway that changes is their cardiac hypertrophic signaling pathway. They're a cardio ager. And we later learned this person's stage 2 hypertensive, so it kind of fits. For this person, basically their immune system changed or metabolic shifted over time. We think as few as 4 time points, you can pretty accurately see how people are aging. And we call these aging patterns ageotypes. So again, everybody's different. And so at the time we were empowered to find 4, we have many more now, but each column here is a different person, each row is an ageotype. And so the people over here, these 4 here, they're aging in all 4 categories. This one on this right box is not much of a kidney ager, but aging in the other 3. This one's a kidney ager, this kidney and metabolic and so on. And there are clinical markers associated with these, so you can actually make recommendations based on this. And I can tell you some of the metabolic agers, they changed their lifestyle. Some of them lost weight or started exercising, and they improved their markers. And I'm not saying they got younger, but they certainly improved their metabolic markers, which we think is very powerful. And I'm going to come back to this theme later. Now, this is done on a rather small group of people, 109, as I mentioned, and Tony Viscoris has done something similar, more cross-sectional for basically 4,000 people and just doing proteomics, and has found a very similar result. Some people will be they'll have a lot of kidney markers in their blood, others heart. Some people are more systemic, they'll have lots of markers. So this idea about personal aging is really emerging as, again, a common theme, and I'll return to it again at the end of the talk. So one question I get asked a lot is, how are you going to scale this? You know, this is great as a research study, which is true. It costs millions of dollars to do what I just said. How are you going to scale this to lots of people? Well, and the answer is I'm in Silicon Valley, so you form a company. And so we formed this company called QBio, which does a medical version of what I just said. It doesn't do the microbiome because we don't know how to act on that yet, but we sort of do now, and we'll probably integrate that. It doesn't do RNA-seq, but it does a lot of other deep measurements. And it also does whole body MRI. So if you ask any person today, any physician, should you do whole body MRI? Nearly 100% of the time they say, absolutely not. And do you know why? Why would you guys not recommend it? You're all physicians, right? By the way, you'll probably never invite me back again. But so well, cost is one, yeah. What else? Not actionable? We'll argue about that. What would you say? Yeah, you always find nodules, right? 100% of the time if you go high enough resolution. So if you ask Mike Snyder, should you do whole body MRI? I say, absolutely you should. And the reason, it's not whether you have nodules or not. We all have nodules, 100% of the time. Just go high enough resolution. The issue is, do you have any growing nodules? And that's where you need the longitudinal measurements. And I can tell you a personal example here where a friend of mine, many of you may know him, woke up one morning Thanksgiving, couldn't move his arm, and they looked at his chest, and sure enough, he had a big tumor on his spine, which they cut out, and they thought they got it all, and everything seemed to work well. And then a month later, they did a whole body MRI, and he had 3 nodules on his spine. And they had no way of knowing if they were there before or if it had metastasized. So you had to treat it as though they were metastatic. Anyway, it was important to know. And I can tell you another example. We just ran a retreat. And you've heard about these. You probably won't like what I'm going to say. But we weren't making money. We let people do a bunch of tests like whole body MRI. You've heard of the Grail test, right? And everybody poo-poos these things. We said, well, all right, come to retreat. We'll talk about these tests. They had to pay essentially what would be the cost of the test. 25 people signed up. It's a longevity retreat, we called it. And one of them was a woman who, from her Grail test, was positive for ovarian cancer. And she was stage 3, it turns out. And she knew it, she found out from Grail because she'd done the whole body MRI. They looked at that, and sure enough, it was there. So you do discover these things. People say, well, it's a late stage test. People would know, but they often don't know, actually. So I do think these tests have value. Now they're really expensive. So we've got to get the cost down cheap. That I agree with. Anyway, here's from the first 100+ people QBio did. They've done over 1,000 now, several thousand. But they did find early ovarian cancer, early prostate cancer, even early pancreatic cancer was picked up by usually by combinations of measurements. And again, these longitudinal measurements are very, very powerful. So we think this is a big deal because I think in the future, these will be 10 minutes. I know they take, and they'll be dirt cheap too. They should be. There's no reason they shouldn't be dirt cheap. So anyway, the point is, I think we can get there, at least for this group of people. It was quite powerful for many, many individuals. So the other thing we've been working very, very hard on is this idea about doing remote monitoring. And so as you heard, we're pretty keen on wearables, and then I'll talk about home microsampling in a minute. So for wearables, there's over 1,000 devices out there. How many of you are wearing a smartwatch or a ring? Oh man, it's a great crowd. Excellent. Well, as you heard already, I wear a lot of these things. I normally have 4 watches. One broke, so I've got it in my backpack. I got to set it up. To my 2 rings here. Even my hearing aids are sensitive. I do wear them for hearing, by the way, but they measure many other things as well. They're actually quite powerful. Some people say they're more accurate. I'm not so sure if that's true. We're evaluating that now, possibly more accurate than what you measure on your wrist. Anyway, what's powerful about these, I'm sure you appreciate, right, is they measure you 24/7 as long as you can keep them charged. So we put them on our cohort pretty early on with the idea they came out as fitness trackers. We said, well, these are pretty powerful health monitors. So we put them on our cohort and discovered right away that you can tell when someone's getting ill from a simple smartwatch, in this case, smartwatch and pulse ox. SpO2 was not on the watches. And this is me. This is when I got my Lyme disease. I was flying to Norway at the time, and you may know your blood oxygen drops on airplanes. If you don't know that, you do now. And you can measure exactly what the air pressure is in an airplane. There's a free app called Barograph. I can tell you I flew here. I think mine dropped to 0.78 atmospheres. You can get it right off the app. And then my case, of course, I track myself a lot with these on the airplane. with a pulse ox and my smartwatch. But anyway, on this particular trip, I was flying to Norway, and I noticed my blood oxygen was abnormally low. Didn't have symptoms yet, so it'll normally drop, but in this case, it was abnormally low. My heart rate jumped up. There were no symptoms. When I landed, it did not come back to normal. And then I did get symptoms off and on, a little bit feverish off and on. And so I went to a doctor in Norway. I warned him it might be Lyme because 2 weeks earlier I was in Harvard, Massachusetts, where 55% of ticks were Lyme infested. And he measures, he sees my monocytes are up and said, yep, you've got a bacterial infection. You should take penicillin. And I said, no, I should take doxycycline. Got a little tense there, as you might imagine. He did give in and gave me doxycycline. And sure enough, it cleared it up. That's the stethoscope here. When I got back, I tested, and sure enough, I had antibodies to Lyme and even had a few antigens. It was a very well-controlled experiment because I had given blood just before I left for that. So I seroconverted during that time. So the point out of all this, that was pre-symptomatically, I could tell when I got Lyme. I won't show you the data for this. You can look it up on paper, but we then looked at the 2 years of data I had to see if we could find other times when there might be suggestions of illness. And sure enough, there were 4 periods. 2 times I had a viral infection. I track all this stuff, even know what the viral infections are. And once I was asymptomatic, but had all 4 times I had high CRP, and you could see that my heart rate had jumped up in advance of symptoms. So we went on to write an algorithm that detects, we called it change of heart, that detects when you're getting ill. And then, of course, along came COVID. This was spring of 2020. And so, and still getting our bursts now and then. If you think about how we measure illness now, we use temperature, which is a 300-year-old concept, and it doesn't work very well for COVID because half the people don't get a fever. I didn't when I got COVID, okay? And you can use antigen or PCR tests, but it's not that convenient. You don't do it all the time because you just don't. It costs money. So we think that smartwatches could be powerful. So when COVID came, we quickly partnered with Fitbit, got 5,300 people enrolled. And step one was see, can we tell when people are getting COVID retrospectively just from a smartwatch? And the answer is we could 80% of the time. We had 32 people who basically were wearing a smartwatch while they had COVID, and they had a diagnosis day and a symptom day. And this is our very first case. This individual, this is resting heart rate here. Let's see if I get that there. And what you'll notice, day 47, it was the day they had symptoms. Day 48, they were diagnosed, but their heart rate jumped up 9.5 days prior to symptom onset. Okay, and it turns out that when we looked at all 32, the median was 4 days prior to symptom onset. We could see this jump up in heart rate. And it turns out COVID is really easy to detect because it was a long pre-symptomatic period. We then went on to write an algorithm that does real-time detection. It's following you in a circadian fashion because your heart rate is circadianly influenced. And so we wrote an algorithm so that if your heart rate's up for too high for too long, you'll get a red alert, basically. Okay, and here's one of our first cases here. This individual had symptoms here on the 21st. They were diagnosed the next day, but they're getting red alerts for 3 days prior to symptom onset. And that's the number with their real-time detection, is about 3 days. 80% of the time, though, there's still a set of people who are missing because they have an unstable background. That's harder to pick up. But nonetheless, what does work, it's quite sensitive. It even picks up, works for Apple, works for Fitbit, should work for all devices. It even works in asymptomatic cases. So meaning, here's a case here, there's a Fitbit case. This individual was diagnosed here on that purple day, but they were getting red alerts for 2 weeks prior to symptom onset. Again, it's very sensitive assays, a little 2 beats per minute, and you pick it up. So it's even more sensitive than antigen tests. We have many examples of this. This is my example where I woke up one morning, that's a real date, April 10th, 2022. I was a little bit congested, and I'm not sure, is it allergies because it's spring? Is it coming down with something? I'm not sure. So I do an antigen test, and I'm negative. That's that T there. See if I can get my arrow up. There we go. I look at my smartwatch, and I'm positive. And I was due to fly to New York City, so what did I do? I got on the plane. Big mistake. Because back then, in order to go to the meeting, you had to get tested. So next morning I tested and I'm bright positive. So I spent a whole week in a tiny little hotel room in New York City, all because I listened to my antigen test and not my smartwatch. Which brings up another point, which is very sensitive, but it's not specific. I didn't know. If I knew for sure it was antigen, although I'm much more cautious now if I get a red alert. It turns out if you look at the COVID negative cases and see what's contributing to red alerts, all kinds of things do, many of which you can contextualize. If you run a marathon, your heart rate will be up for several days. That'll trigger red alerts. But the number one trigger of red alerts is workplace stress, actually. So it's a mental health detector as much as a physical health detector. And I think that's really true. If you wake up first thing in the morning and you've got red alerts, and there's not an obvious cause for it, you're probably either mentally or physically stressed. Something's off. And so we would argue this is powerful. We're actually trying to do a lot with mental health detection now for anxiety and depression using devices. So we think this is the tip of the iceberg. If you think about smartwatches, they're shining light into your blood. They're measuring a lot of stuff. And we can, this was the 2016 watch. We're repeating it now with a better one. But basically, you can use machine learning and AI to see if you can predict clinical variables from basically the signals from a smartwatch. Take features of a smartwatch and predict these various things. And it actually works pretty well for red blood cell count, hemoglobin, hematocrit. Even can pick up hemoglobin, A1C, and fasting glucose from values, because one of the things, smartwatches, they're quite good for heart rate variability. They're different quality, I'll say, with skin temperature. But one thing they measure is electric conductance on your skin, GSR, which turns out to be valuable for diabetes, because when you're diabetic, you get dry skin. I know that personally. I'm a type 2 diabetic, a weird one. We'll get to that perhaps in a minute. But the point out of all this is that you can pick up these values from a smartwatch. They're not clinical grade measurements, but they're plenty good to see a shift from your baseline, which is then we just need alerting systems for this. The other area we've been pushing very, very hard is this idea about continuous glucose monitoring, following people's glucose levels. So how many of you have worn one of these CGMs? A few. They're pretty powerful, right? You'll never eat the same again, I think, once you wear one of these. I mean that in a good way, actually. So, so these things, they measure glucose every 5 minutes. And what we did, they were being used a lot for insulin-dependent type 1 and type 2 diabetics. What we did was we started throwing them on so-called normal and pre-diabetics. And we discovered a lot of so-called normal people. Some are normal and have pretty good glucose control, like this top one here. Some are moderate glucose spikers, and some are severe glucose spikers, just as bad as diabetics. So we classified people into what we call glucotypes based on their spiking in glucose levels. And then we went on and discovered that different people spike to different foods, as did Aaron Siegel. And so some people spike to bananas, some to pasta, some to white bread, some to brown bread. We're all different. And Aaron went on to show about 21% of that's due to your microbiome. Here's a recent paper we just published where we had 55 people each eat 7 different carbohydrates, but the carbohydrates are in different forms: beans, berries, bread, grapes, pasta, potatoes, and rice. They did it in duplicate or triplicate. And most people are like this. They're rice spikers, okay? You can see this here. And in fact, rice is worse than ice cream for most people. I don't realize that, but it's true, okay? This person's a bread spiker. See if I can get my arrow back. The next one over here we go is a grape spiker, pasta, potatoes. What's going on? We think this reflects the heterogeneity of type 2 diabetes. So it involves your liver, it involves your pancreas, it involves your muscle, even your brain is a major glucose consumer, if you will. And so there are many signaling pathways. There's insulin, the incretins, there are glucagon, right? All this, there's a lot of complex biology here. Talk to Ernesto, and probably have to talk to him for about 3 weeks to figure all this stuff out. But The point out of all this is that it's very complicated. And diabetes, we lump as type 1 and type 2, but we think type 2 has many subtypes, even type 1 these days, probably more. So what we did with Tracy McLaughlin is we went in, and these are people ordered on their hemoglobin A1C on the left. See if I can get this up. The blue is so-called normal. Yellow is pre-diabetic, and a diabetic at the bottom snuck in the study. Here we go. Now I got it. And we typed them for muscle insulin resistance, beta cell defect, incretin defects, and hepatic insulin resistance, all based on OGTT. Again, this, for those of you who follow this, you'll know what I'm talking about, SSPG assays, and even an intravenous glucose injection. And the bottom line out of all this is that you'll notice everybody's different. So let's see, 2 examples here. This person is muscle insulin resistant, normal for incretin and intermediate for others. This person's an incretin defect, insulin sensitive and intermediate for beta cell. So the point is, just look at the pattern. Nobody's the same. We're all fairly different. So you might say, so what? Oh, let me tell you one more thing. Just from the shape of a curve, and now with machine learning, we can actually tell your subtype pretty well for muscle insulin resistant and beta cell. Not so good yet for the other 2. We're working on that. And that's using features from the curve. You see everybody, if you've ever if you looked at people who have these, basically everybody says sharp rise, slow fall. You can see all kinds of patterns there. And so again, from features on a curve, you can actually predict and even get a better prediction of people's resistance, of their muscle insulin resistance and beta cell from a home CGM than you can from an OGTT because of high resolution mapping. So it turns out, again, we can make predictions from a CGM curve. Well, that's nice because then for $50 and drinking a shot of glucose, you can tell somebody's subtype versus many hours and thousands of dollars you would spend in a clinic now. All right, so what? Why do I care about my subtype? Well, the answer is it determines the foods you'll spike to. So this is muscle insulin resistant. This is now aggregate signal over all participants. But if you're muscle insulin resistant, you'll be a potato spiker and a pasta spiker, but not if you're sensitive. Similarly, if you have a beta cell defect, you'll be a potato spiker. We're still sorting out more of this, but the point is you can match people's subtype to foods. We're even taking this one step further. This is still in progress, but you've probably been told you should always eat your salad before your French fries, right? That's generally true. So eat something before you eat these spiky carbs. And we tested what we call mitigators, protein, you'll see here, fiber or fat in these forms here. They would eat 10 grams of this prior to eating white rice, which again spikes most people. And then we looked at what would suppress the spikes. And it turns out if you're normal, fiber and protein will suppress basically those spikes, but if you have a beta cell or it turns out muscle insulin resistant, it doesn't work. So the mitigation depends on your subtype too. And as I say, we're still sorting that out. We can take this then we formed a company around, I've conflicted in everything I'm telling you, by the way. You probably figured that out. All right. So anyway, this company actually takes your lifestyle, has AI models and predicts people's, spiking, and people who do the program actually both lose weight and improve what's called their time in range. So here's how we've taken this one notch further. We basically had people wearing a CGM, 30-some, while they were subtyped. They're wearing a CGM and a smartwatch and food logging and tracking activity, et cetera. And then what we do is we build AI models to predict the effects of their activity on glucose levels by hemoglobin A1C or on their subtype. And so what you'll notice, starts simple. Here's A1C. A lot of this is known already. Eat your biggest meal first thing in the morning, you'll have lower glucose. Late at night, higher glucose. Starchy vegetables, higher glucose. Fruits, lower. Most people don't sleep enough. I'm sure if that's true for physicians. Sleep more, lower glucose. Okay. So you can even do this for subtypes. So if you're muscle insulin resistant, exercising more will lower that. And this is very relevant for me. I said I'm a type 2 diabetic. I'm a beta cell defect. I actually got there after I'm high on the polygenic risk score. I'm one of the few people that worked for. But I became insulin resistant after an RSV infection. And sorry, I became diabetic, I should say. It turns out I'm a beta cell defect. And so I shifted, you can't tell, I was a runner, but I shifted from running to weightlifting. I gained 10 pounds of muscle mass. I do whole body MRI. I've done 22 of them in the last 9 years. So and I gained all this muscle mass, had zero effect on my glucose. And the reason is I'm a beta cell defect, right? I'm not muscle resistant. It doesn't matter how much muscle I have. It's not going to help me. So this is why we think knowing your subtype. And it turns out I'm a metformin non-responder on top of that. And I wind up responding well to this drug that I can never pronounce right, but Ernesto will tell you, rapinolide, but rapilonide, there you go. All right, so I do respond well to that, and that promotes release from your pancreas. And then these days, like everyone else, I'm on GLPs because they work really well, but it does have other issues. Yeah, I'll leave it at that. Here's the last set of stories I think I'll tell you about, which is this home microsampling. The idea, oh yeah, I see we're running out of time. All right, maybe I'll go through this kind of quickly. But the idea is, why do you go to a doctor to get measured? The consequence is we don't get measured very often, and we should really be measuring, we think, a lot more often. And so we spent 7 years perfecting this and home microsampling. Start with these devices. Now we use these patches on your shoulder. Basically, you collect droplets of blood, overnight it to the lab, and now we measure 7,000 analytes using omics profiling. And the net result is that most analytes are stable, and then you can do fun experiments. And here's one here. We had 32 people drink this shake called Ensure Shake, and basically they were fasted. We measured them before and various times afterwards. Hundreds of molecules shifted, and we can measure things like insulin, all kinds of important molecules. The cool result is here, everybody reacted to the shake differently. So this person, each box is a different person. This person, their carbs dropped. For these people, it went up, relatively speaking. This person's flat, this one dropped, that one dropped. Informatically, we can group people into 5 classes of people. So for this group of people, the relevant features are going to be the brown, that's your inflammatory markers. Gray is the average. For this group of people, their inflammatory markers goes down when they drink that shake. For this group of people, it goes up. Same with this group. Same shake, pro-inflammatory on some, anti-inflammatory on others. 10% of people have irritable bowel syndrome. We could actually measure it with this kind of an assay. So we think it'll be very, very powerful. There's one person who gave a sample every hour, and the idea was to track activity with, basically what people do with their both, biochemical and physiological responses. And the net result is, so they basically gave 100 samples over 7 days. And you can start making associations, again, between people's activity, what they do, as well as when they do it, with various markers. And so some are obvious, like your insulin C-peptide goes up in response to glucose. But here's an interesting one. Alpha-synuclein involved in dementia and Parkinson's shows a very interesting pattern we think correlates with stress. And so the net result is that we think the hypothesis is that if we can track this and tell people to minimize this, perhaps we can push off them getting dementia. That's the hypothesis. I'll skip over this, but we have a dashboard to bring in all these data. This is where I'd like to see this go, where you would use big data, including genome sequence, probably get your genome sequence before you're born. And then together with biochemical measurements, a wearable, we're all going to have personal health agents tracking our health in the future. It's already happening here. And then from this, you can predict your risk, what you're catching, catch disease early, and so on, and treat it. Once again, we do spin this off. So we form lots of companies around this kind of work, actually 18 now over the years. I want to tell you about this one. This is the microsampling company where you put a patch on your arm, you collect blood, you mail it in, and they'll measure 650 metabolites spanning 20 wellness categories. So things like metabolic health, oxidative stress, inflammation, and they'll even measure your biological age. And this brings us back, they can deduce your ageotype. Okay, so this person, their biological age is older than their chronological age, so 53 versus 51. But you break it down, the ageotypes, their inflammation's fine, metabolic age up a bit, heart age much, much higher. And so use AI with their data along with all the scientific literature that's out there and make very specific recommendations about what they should do. And 95% of people improve their markers. Again, I think this is how we're all going to use AI agents in the future. Probably have to have them lock your fridge and things like that too sometimes. But anyway, that you have to wire your house then. We're not quite there yet, but you get the idea. Because I've run a little late, I'll just say I know I've overloaded you with a lot of info, but we use genetics and genomics along with all these big data collection methods, especially wearables, for trying to better manage people's health. Here is the people who did the work, and I want to save a few minutes for questions, and I'm sorry I'm shortchanging them, but anyway, that's my story.
Wow, Michael, you blew us all away. That was an amazing grand rounds. Thank you so very much. There's lots of questions. I don't know who's online, but I've got about 100. But I'll start with just maybe one or two. So, what do you think is the main obstacle, the main barrier for this to sort of become available to everyone? Is it cost? Is it ethics? Is it the FDA? I mean, what is preventing everything from sort of coalescing in the direction that you're going in?
Who pays? Nobody pays to keep you healthy in today's world, right? Most people get paid. Most people visit doctors when they're sick. We need to financially incentivize people, get the incentives aligned. So I think there's only two things that motivate people: money and family. So I think you should get a discount if you're wearing a smartwatch because they guarantee you will add more steps on. You should get a discount in my world to if you got your genome sequence because you'd know what you're risk for and you'd get screened for those things, much like that list I showed of the Mendelian cases. So I think we need to do this. The only place I've seen this financially align is there's a life insurance company, John Hancock, that has a vitality program. They give you points if you wear a watch because there the incentives are aligned. If you live longer, they make more money. Whereas in the healthcare system, that's not necessarily the case. So we've got to get those things aligned, and I think that will do the trick. So I think when you join, University of Miami healthcare system, you should get a watch for free, right? They're going to be $50. A $50 watch can do everything I told you about disease detection that an Apple Watch can, quite frankly. Apple Watch has more bells and whistles, but.
Mike, fast forward 40 years, how do you see the physician-patient relationship going to be when you come to see, a doctor?
Yeah, a couple things. First of all, I think a lot of the information will be entered before you see the physician. So your watches should automatically be brought in. Concierge services are already doing this. Most healthcare systems are not bringing these kind of data. They're way more accurate than what you measure in a doctor's office. I think you and I may have talked about this. A physician always asks, or the nurse says, so when did this start? All you have to do is look at your watch, you know exactly when it started. You just look for the jump up over your baseline. So I think we should be bringing a lot of this information automatically. The questionnaires can all be automated. I don't know if this happens here, but even at Stanford, been in the emergency room from bike accident, other things. First thing to do is they measure your blood pressure, a few other things. You go to the next room, they measure your blood pressure, a few other things. You go to the next room, they measure your blood pressure. You measure it 3 times. It hasn't changed in my case at all. Why isn't all that stuff automated and brought in? It should be. We should be bringing this stuff in automatically. And I think back to your point, the wearables we can integrate now and should be integrated now that you'll better manage people's health. So we waste in the 30-minute doctor's appointment, I would say the first 10 is by the nurse asking questions. Well, that stuff can all be automated. You don't need a nurse to put on a cuff to measure blood pressure, or maybe one can watch 50 people put on cuffs all at once. Why do we spend a lot of time on things that that could easily be collected and probably more completely collected if you let the patient enter it. And when they need health, then you have the nurses, they can manage multiple people at once. And I think you'd give better care, try and get more contact time with the physician. In the future, we all have to have AI agents. I have multiple agents on me, as you might imagine, and they make recommendations that, and I have a lot of people who look at me and make all kinds of recommendations. And my AI agent made two that nobody else spotted, one of which is probably correct. I should be taking more zinc to up my monocytes. And another is controversial, but nobody else had flagged that. It's on SGLT2 inhibitors for my diabetes. So anyway, the point out of all this is we should have those all pumping in, and that'll be automated. And there are beautiful systems out there now. And you may know that, doctors alone have a certain accuracy. AI is always higher, usually for most assays. And AI plus doctor is worse, actually. So I'm not saying we shouldn't have doctors looking over the AI. We should. We just have to learn how to use AI better. And so that's how it's going to be in the future. Your agent will have a whole bunch of things that it's flagged. Your physician can still override and read all this, but it pulls in information that no one would know. And I can give you many examples if you want to hear a fun one, which is a friend of mine built with vibe coding, just built a system and said, I want to look at supplement interactions. Then he loads up his supplements, one's zinc and one's magnesium. And the AI said, well, you got to take them 6 hours apart because they use the same transporter. No physician's going to know that, right? I wouldn't know that. But AI pulled it from the literature. You can look it up, you click on it. That's the key to verify. And sure enough, the AI was right. So I think we're going to get a lot of information like that. Again, that no human can process in short periods of time. And that's the power of this in the future. Now, you have agents talking to other agents. So the accuracy has really skyrocketed on this stuff.
Thank you, Professor Snyder. I'd like to borrow a few of your agents to look like you in terms of my weight, but you're amazing. And on behalf of the Department of Medicine, I want to present you with our Eugene Safety Annual Research Day Keynote Speaker Award, and we'll get a picture of you in just a minute. But thank you so very much, Professor Snyder. I want to remind everybody that at 2 p.m. on the Science Quad, we're going to have all the posters displayed, over 150 of them, 174 to be exact, and they'll be up until 4 p.m., at which time we'll have our award ceremony, and everyone's welcome. As we heard earlier, there'll be refreshments there as well. I want to remind everybody the QR code for the MEC and CME and MOC credits, and wish you all a wonderful day and hope to see you later this afternoon on the quad. Thank you. This is great. I'm blown away.
It went so long.